Assalamualaikum warahmatullahi wabarakatuh.
Hello everyone!
We are from Group
3.
Consists of
Ashifa Iman Maharani.
Jihan Reihana, Mediana Fauzia, Muhammad Faisal.
So we are from IPSE 2020 from Indonesia University of Education. So in this structure and function of Glenn's course, we will record our learning activity. So happy watching!
Alright, for the objective of this experiment is comparing the stem, leaf, and root of Dicot and Monocot.
For the tools and material, there are monocular microscope and egg slide consists of lilium leaf, geamice, cucurbita, geamice root, geamice stem, cucurbita stem.
Okay, let's do the observation of the object by using the microscope. So this is the slides, and our group mates was observe the object by using the microscope. Let's see by using the camera.
Well, the next is results. So the result that we got from these observations is lilium transversal leaf. It is classified as monocotyle. And the structure that exists in this plant are sclerenchyme, upper epidermis, stoma, bulliform cell, vascular bundles, and chloroplast. The second that we got is a zeamice transversal stem. It is classified as monocotyle, and we can see there are some structure which are phloem and also xylem. The third is cucurbita transversal section stem. It is classified as dicotyle. And there are some structure that exist in this plant which are phloem, parenchyme, xylem, collenchyme, cambium. The fourth is zeamice transversal section root. It is classified as monocotyle, and there are some structure which are epidermis, endodermis, cortex, phloem, xylem, and pericycle. The fifth is Ficus transversal section leaf. It is classified as dicotyle, and we can see there that exists in this slide. There are some structure which are upper epidermis, palisade layers, vascular bundles, spongy layers, and the last is lower epidermis. The sixth is zeamice longitudinal section stem. It is classified as monocotyle, and we can see there from the structure which are fibers with parenchyme, xylem vessel, and phloem. The seventh is cucurbita longitudinal section stem. It is classified as dicotyle, and we can see there are some structure that we can observe. The first is fibers, scalery worm thickening, xylem, parenchyme, product xylems, vessel elements. The last is helianthus transversal section old root. It is classified as dicotyle, and we can see there the structure that is exists from this plant, which are endodermis, xylem, root hairs, phloem, and cortex.
Well, let's see the discussion from this lab report. Alright, so this is our lab report in Canva. So let's see the discussion. So there are some characteristics of monocot and dicot, and based on our research, we have 8 slides. And for the first is lilium transversal section leaf. This leaf is categorized to monocot in transverse section, and the palisade layer is about 200 nanometer thick. And the vascular tissue is surrounded by the large parenchyma cells, which help to support the leaf by their turgidity. A smaller vein can be seen toward the right of the section, and the several guard cell pairs are visible in the lower epidermis and the stomata, leading to large substomal air spaces and allowing gas exchange with the mesophyll tissues. The second is the stem of zygomates in transversal section. It exhibits swallowing tissues from outside. Within the monocot stem has vascular bundles near the outside edge of stem. Vascular bundles are scattered in parenchymatous ground tissue, and there is no pit region in monocots, and dicot stem have bundled inner ring surrounding the parenchyma cells in a pit region. In a monocot stem of zygomates, there are epidermis, hypodermis, ground tissue, vascular bundles, and phloem.
For cucurbita transversals, sectional stem showing light shape, tubes, and vascular, it means the stem categorized as a dicot. Every stem has same form, which is like pentagon. The structure of transverse section of a young stem of cucurbita has some structure which are epidermis, vascular bundles, collenchyma, parenchyma, sclerenchyma, structure sheath, pit or hollow, and leaf structure bundle. And the fourth is cucurbita longitudinal section stem categorized as dicot. Cucurbita longitudinal stem show the scarified parenchyma, primary metaphyllum vessel element, protoxylem vessel element, scalariform thickening, and reticular thickening. In this stem, which has a xylem that has function as apoplastic conduction of water and solute, mechanical support, and storage of water and nutrients.
Next one, because the S leaf is categorized as a dicot. Generally, the leaf consists of the same anatomical structure, such as epidermis, stomata, mesophyll, and vein. Ficus has a litosis cell, which is a specialization from the epidermis cell located in the leaf. Ficus is also a plant that has an epidermis multilayer. Epidermis multilayer is from protoderm from pericranial division. It was caused by there are so many stomata under the leaf. Stomata is the most important thing for plant life, especially in photosynthesis. And the next one is the maize TS root, which is categorized as a monocot. This is indicated by the separation of phloem and xylem, or what is also called closed collateral. The arrangement of the phloem and xylem is a circle. Root of zygomates has phloem, pericycle, endodermis, metaxylem, cortex, pith, and protoxylem.
Okay, for the next is zygomates longitudinal section stem, and it's categorized as monocot. This is indicated by no vascular cambium found in its structure, and zygomates stem has metaxylem, xylem parenchyma, protoxylem, sclerified parenchyma, bordered pith, and fibers. And for the last is Helianthus transversal section old root. This plant categorized as dicotyl. This is indicated by the xylem tissue that appears like a four-pronged star. The tissue between these branches is the phloem. This plant also has root hair, which is only dicotyl root has. This plant has epidermis, phloem, endodermis, and cortex. Helianthus old root have formed pith rays formed by the cambium in the first year. Xylem consists of protoxylem and metaxylem, and is surrounded by a sclerenchyma sheath. Phloem consists of sheath cells and conducting cells. This is an intervascular cambium. The cortex is filled
So, the
with
conclusion is based
parenchymatous.
on our experiment result, the monocots consist of lilium and zymase, and the dicot consists of cucurbita, ficus, and helianthus. Every structure of plants has their own characteristics related to their own functions. Meanwhile, the differences between monocots and dicots are they have own characteristics. So the characteristics of monocots are vascular bundle scattered, root xylem and phloem in a ring, and leaf veins form a parallel pattern. While the characteristics of dicots are vascular bundles in a ring, root phloem between arms of xylem, and leaf veins form a net pattern.
So that's all from our activity.
Thank you for watching.
See you in the next video.
Bye.